The organelles. Most are membrane clone structures having the same components as plasma membranes. They do not float freely within the cytosol but are interconnected and joined by the framework established by proteins of the cytoskeleton. Members of these collections of organelles have a characteristic arrangement within the cell and proximity to each other that allows them to carry out their function in protein processing. Some other organelles are involved in digestion and detoxifications. All are important, especially in protein synthesis. Some of the organelles are membranous organelles such as endoplasmic reticulum, Golgi, lysosome, and peroxisomes, while the other are the non-membranous organelles like ribosome. And although they play a part in protein synthesis, each has different functions. First is the ribosomes. Ribosomes are minute particles consisting of RNA and associated proteins that function to synthesize proteins. Proteins are needed for many cellular functions such as repairing damage or directing chemical processes. Ribosomes can be found floating within the cytoplasms or attached to the endoplasmic reticulum. The endoplasmic reticulum. In essence, the transportation system of the eukaryotic cell, and has many other important functions such as protein folding. It is a type of organelle made up two subunits, rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough endoplasmic reticulum RERs are series of connected flattened sacs, part of a continuous membrane organelle within the cytoplasms of eukaryotic cells that plays a central role in the synthesis of proteins. While a smooth endoplasmic reticulum or smooth SER is a membranous organelle found in most eukaryotic cells. Its main functions are the synthesis of lipids, steroid hormones, the detoxification of harmful metabolic byproducts, and the storage and metabolism of calcium ions within the cell. This is the Golgi apparatus, also known as the Golgi complex, Golgi body, or simply the Golgi, is an organelle found in the most eukaryotic cell. Part of the endomembrane system in the cytoplasm, it packages proteins into membrane-bound vesicles inside the cell before the vesicles are sent to their destination. Next is the lysosome. A lysosome is a membrane-bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. They break down excess or worn-out cell parts. They be used to destroy invading viruses and bacteria. Next is the peroxisome. A peroxisome is a membrane-bound organelle formerly known as microbody, found in the cytoplasm of virtually all eukaryotic cells. Peroxisomes are oxidative organelles. Frequently, molecular oxygen serves as a co-substrate from which hydrogen peroxide is then formed. Translation is the synthesis of a protein in the cytoplasm using an mRNA template that has been exported from the nucleus. For protein biosynthesis, the mRNA molecule binds to protein RNA complexes called ribosomes in the cytoplasm, where they are translated into polypeptide sequence. The ribosomes is another one of these RNP machines that mediates the formation of polypeptide sequence based on the mRNA nucleotide sequence. Initiation of translation begins when a small subunit of the ribosome attached to the methylated cap at the pipe end of the mRNA then moves to the translation initiation site. Another key molecule is the family of transfer RNA molecules, which carry anticodons complementary to the mRNA codons. The first codon to be translated in the mRNA is typically the AUG, and the tRNA that corresponds to this carries the amino acid methionine. The large ribosome subunit now binds to the assembly, creating the peptidyl or P site and aminoacyl or A sites on the ribosome. The first tRNA occupies the P site and the second tRNA complementary to the second codon enter the A site. As peptide elongation continues, the growing peptide is always transferred to the tRNA at the A site by being peptide bonded to the amino acid carried by that tRNA. Then, the ribosome then moves along the mRNA so that the tRNA carrying the peptide synthesized to date remains at the same place on the mRNA, held by the hydrogen bonding between its anticodon and the mRNA codon, but is shifted to the ribosome's P site by movement of the ribosome. Synthesis is initiated at the amino terminal end and ends at the carboxy terminal end of the polypeptide. When a stop codon is encountered in the A site, a release factor rather than tRNA enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed polypeptide is released and folds into a functional three-dimensional protein molecule. Protein post-translational modification or PTMs increase the functional diversity of the proteome by the covalent addition of functional groups or proteins, proteolytic cleavage of regulatory subunits, or degradation of entire proteins. These modifications include phosphorylation, glycosylation, ubiquitination, nitrosylation, methylation, acetylation, lipidation, and proteolysis, and influence almost all aspects of normal cells biology and pathogenesis. Last process is also called the protein targeting or the protein sorting. During transit through the cytoplasm, proteins called chaperones become associated with a newly synthesized polypeptide to modify its folding and deliver it in a suitable form to the proteins of translocator complexes embedded in the organelle membrane that import it into the organelle. When it is delivered, a complex called the signal peptidase removes the signal sequence. So although the coding for this sort of signal sequence must be contained within the mRNA sequence, it doesn't appear in the final working protein. Most newly made proteins in the ER lumen or membrane are incorporated into small transport vesicles. This is either fused with a cis Golgi or with other to form the membrane stacks known as a cis-Golgi reticulum network. From the cis-Golgi create proteins, mainly ER-localized proteins, are retrieved from the ER via a different set of retrograde transport vesicles. In the process called cisternal migration or cisternal progression, a new cis-Golgi stack with its cargo of luminal protein physically moves from the cis position to the transported position, successively becoming first a medial Golgi cisterna and then a trans-Golgi cisterna. As this happens, membrane and luminal proteins are constantly being retrieved from later to earlier Golgi cisternae by smaller retrograde transport vesicles. By this process, enzymes and other Golgi resident proteins come to be localized either in a cis or medial or trans-Golgi cisternae. Proteins destined to be secreted move by cisternal migration to the trans phase of the Golgi and then into a complex network of vesicles, termed the trans-Golgi reticulum. From there, a secretory protein is sorted into one of new types of vesicles. These proteins are sorted into trans-Golgi network into transport vesicles that immediately move to and fuse with the plasma membrane, releasing their contents by exocytosis.